Start the class, madam. Madam, please start the class.
Hello, am I audible?
Yes, madam. Clear. Please start the class, madam.
That's what I'm starting, sir.
Okay.
So myself, Kirti Rai. I'm working as TL in Zoology, Tsrjc Sarvail. And today we are going to discuss about the human reproductive system. And in this chapter, we will be learning about all the events of reproduction. So first of all, when it comes to the reproduction, first of all, what is reproduction? Reproduction is nothing but the process of production of young ones of its own type. That is, when an organism reproduces, it produces a new organism, that is offspring, or also known as young one, which is exactly, it can be similar to its own type, or it will be different from its own. And when we talk about reproduction, there are mostly two types of reproduction: asexual reproduction and sexual reproduction. So asexual reproduction, it is seen in the lower organisms, or we can say the primitive living organisms, such as protozoans, cnidarians, coelentrates, platyhelminthes. And in the asexual reproduction, it involves only one parent cell. There is no need of two parent cells in the asexual reproduction. And the organism which is produced here in the asexual reproduction will be exactly similar to that of its offspring. And the other one, which is sexual reproduction, it involves two organisms, that is male and female organism. Now, when two organisms are involved, the offspring which will be formed is definitely not going to be similar to, exactly similar or identical copy to that of its parents. It will be having different characteristic features, and it shows variation from that of its parents. And in the sexual reproduction, male and female gametes are required. So that is about the basic knowledge about the sexual reproduction in the organisms and the asexual reproduction. Now here in this chapter, we are only going to discuss about the reproduction in the human. So reproduction in the human, we all know that it involves sexual type of reproduction. Now, in the sexual type of reproduction, there are different processes which are seen in the human. Now, these reproductive events are on the slide. You can say they are in the order, that is, it starts from the process of gametogenesis, and then the next step is insemination, fertilization, implantation, gestation, and parturition. Now let us discuss about each of them separately. So what is gametogenesis? Gametogenesis is nothing but the formation of gametes, that is sperm, which are sperm and ova. The gamete, the sperm is a male gamete and ova is the female gamete. The sperm is produced by the process of spermatogenesis and the ova is produced by the process of oogenesis. Now, first of all, these gametogenesis will be the ones which are going to produce male and female gametes. About this gametogenesis, spermatogenesis, and oogenesis, we are going to discuss in detail in the coming chapters. And the next step is insemination, that is transfer of sperm into female genital tract. So that is the second step which is present in the reproductive events in the humans. Then after the process of insemination, the next step is fertilization. Fertilization is nothing but fusion of male and female gamete, that is ova and sperm, in the female reproductive system. So because it is occurring inside the female reproductive system, the type of fertilization is internal fertilization. And the formed zygote, will be diploid in nature, whereas the male and female gametes are haploid in nature. The next step in the process is implantation. Now, after the formation of the zygote undergoes cleavage, that is cell division, which will be a mitotic type of cell division, and it develops to form a blastocyst. Now, the attachment of the blastocyst to that of uterine wall is known as implantation. The next step is gestation, which is nothing but fetal development period from the time of conception until the birth. The full gestation period is normally for 9 months in the humans. And the last step is parturition. Parturition is nothing but delivery of the baby or the birth of the organism, or it is also known as the action of giving birth. So these are the reproductive events which are seen in the humans. Now let us discuss in detail about each and every event and how does it start, and let's discuss about everything which we are going to deal here. Now the first thing is, here we are going to discuss about the male reproductive system. Now, male reproductive system, the reproductive parts which are present in the male are there, and there will be two type of things: sex organs and the male accessory genital glands. Now, the sex organs in males, it includes a pair of testes, a pair of vas efferentia, a pair of epididymis, a pair of vas differentia. Now, the testis is the primary sex organ, and the vas efferentia, epididymis, and vas differentia, these are the accessory ducts which will be helping the sperm to move. The urethra and penis are the organs for transfer of sperm. And the male accessory genital glands are nothing but, there are three male accessory genital glands: the seminal vesicles, which is a pair, prostate gland, and a pair of bulbourethral glands. So these are the these are the parts of the male reproductive system. Now let's discuss about each of them in detail. So in the next slide, you can see here, is also known as testicles, which is a similar. Now, in every human being, in every male, there will be a pair of testes. Means two testes will be present, and these testes are pink, oval in shape, and they are pinkish in color. And now these testes are suspended in a pouch which is known as scrotum. So if I enlarge this diagram, you can see here there is a structure which is known as scrotum. Now this scrotum is the outermost thing in which the testis is descended, and this scrotum helps in maintaining the testes are suspended in the abdominal cavity within a pouch called scrotum. Now, what is the role of scrotum and why this testis cannot be there in the abdominal cavity? Why it has to be suspended into the scrotum is given here. The scrotum helps in maintaining the lower temperature of the testis than the normal body temperature. Now, why the testis needs to have lower temperature when compared to the normal body temperature is, now the testis is nothing but a primary sex organ which is finally going to produce the male gamete, or we can say sperm cells. Now these sperm cells will be produced by the process of spermatogenesis. Okay. Now this process of spermatogenesis, the process of spermatogenesis requires lower body temperature than the normal body temperature. So in the scrotum, the testis will be that is the reason scrotum. And this testis will be descended into the scrotum.
Please, sound please. Sound please.
Full sound is there, ma'am. Hello?
Okay, carry on.
Okay. Now, the temperature of the test scrotum will be lower to 2 to 3 degrees Celsius lower than that of the abdomen. Now, if the testes do not descend into the scrotum, then sterility will occur because the normal production of the sperm is inhibited as it needs less temperature than the body temperature. So, because of that, the male germ cells will not be able to produce, that is process of spermatogenesis will not occur. So if there are no spermatogenesis process, we cannot get the male germ cells, so which leads to the sterility in the human. Now the failure of testis to descend down into scrotal sac is called cryptorchidism, the condition in which the testis is unable to descend into the scrotal sac. That condition is known as cryptorchidism. Okay. And the next thing is this testis is enclosed in a fibrous envelope which is known as tunica albuginea. You can see here the tunica albuginea which is enclosing the testis. And this tunica albuginea extends inwards and it divides the testis into testicular lobules. You can see in the diagram, these regions are nothing but the invaginations of the testicular, invaginations of the tunica albuginea. Now these invaginations will divide the testis into different testicular lobules. Okay, nearly 250 testicular lobules are present in each testis, and each lobule contains 1 to 3 highly coiled seminiferous tubule. So this is, if this is one lobule of the one testicular lobule, in this testicular lobule there will be 1 to 3 highly coiled seminiferous tubules will be present. Now these seminiferous tubules are the ones which are going to be forming the male germ cells. So the main function here is inside the testis is of the seminiferous tubule. Now let us discuss in detail about the structure of seminiferous tubule in the next slide. Now you see here the seminiferous tubule. The seminiferous tubule is lined by germinal epithelium. Now you can see the germinal epithelium here on the outer side of the seminiferous. This is one seminiferous tubule, and you can find the germinal epithelium. If we look here in this one, this part is the germinal epithelium. Now this germinal epithelium is the one lining the seminiferous tubule. This seminiferous tubule consists of the following parts. It consists of two parts, the male germ cells and the nourishing cells, which are also known as Sertoli cells. So the male germ cells are the spermatogonial mother cells, which are finally going to form the sperm cells, or we can say the male gamete. And nourishing cells are the Sertoli cells, which will be providing the nourishment. This part, this one is the Sertoli cells, and these are the male germ cells. You can see here the spermatogonia. And in between the seminiferous tubule, there is another part which is known as interstitial space, or in this interstitial space, there will be interstitial cells, which are also known as Leydig cells or cells of Leydig. Now, what is the role of these three things, which we are going to discuss in the next slide. Now, all these seminiferous tubules finally they will be opening into a structure which is known as vas deferens. Okay. Now, before going to this, let's discuss some points. Now, where the vas deferens will be originated and how it enters into the other part. Now, if you see this part, the seminiferous tubule, all the branches of the seminiferous tubule will be finally opening into a structure which is known as rete testis. You can see here all the seminiferous tubules are opening into a duct-like structure which is known as rete testis, and these rete testis will be opening into the efferent vas efferents. These efferent ductules are nothing but the vas efferents, and these vas efferents will be opening into the epididymis. This part. Now, the epididymis, head of the epididymis will be finally opening into the vas deferens. Now, there is a structure which is known as inguinal canal, which is nothing but a canal which connects. You can see here inguinal canal. This is a canal which connects the scrotal sac, the cavity of the scrotal sac, to that of the cavity of the abdominal. That is, abdominal cavity is connected to the cavity of the scrotal sac through this inguinal canal. And this inguinal canal will be, this is the structure through which there will be many various structures from the scrotum will be opening into the abdominal cavity through this inguinal canal. Now, this testis is connected to the wall of the scrotum by a structure known as, or we can say the testis is held in position. This testis is held in position by a structure known as gubernaculum. This gubernaculum will be connecting the wall of the scrotum to that of the testis. And the other things, that is the inguinal canal, which is connecting the scrotal cavity of the scrotal sac to that of the abdominal cavity, it will be carrying different structures such as the blood vessels of the blood vessels which are coming to the scrotum, or which are going to supply blood to the testis and taking away the blood from the testis, that is spermatic artery and spermatic nerve, will be coming from the abdominal cavity through this inguinal canal, as well as the vas efferens will be also going out through this inguinal canal to the abdominal cavity. And then there is a structure which covers completely, or which is present completely outside to that of the testis, that is outer covering of the testis, is known as tunica vaginalis. This tunica vaginalis is the peritoneal layer. It is the peritoneal layer. You can see here, tunica, this part, tunica vaginalis. It is covering the testis, or we can say the outermost membrane of the testis. So when we go from outside to inside, what we find is the outermost is the scrotal sac, and inside the scrotal sac there is outermost layer which is tunica vaginalis, which finally this tunica vaginalis, below this tunica vaginalis there will be the layer which is tunica albuginea, and this tunica albuginea is dividing the testis into different lobes, and finally opens into the seminiferous tubules. So this is the structure of scrotum and testis, inguinal canal, and all the organs which are present here. Next, as we have discussed in the seminiferous tubule, now let us discuss each and every part of the seminiferous tubule. Yes. So as we have seen, the seminiferous tubule consists of spermatogonial mother cells or the male germ cells. This spermatogonial mother cells, if you see the testis here, this seminiferous tubule, if we take a cross section of this, then we are enlarging this part. Then what are the things which we find here? We can see here. Here we have the spermatogonial mother cells, or which is also known as spermatogonia, and this is diploid in nature. Means the number of chromosome is 46 here. And in between them, in between the spermatogonial cells, we can find the Sertoli cells. These Sertoli cells will be providing the nutrition to the developing spermatogonial cells. Now, these spermatogonial cells will undergo first mitosis division to form the primary spermatocyte. This primary spermatocyte is also a diploid, that is 2n. The number of chromosome is 46 here. And this primary spermatocyte will undergo first meiotic division. It will undergo the meiotic division. This spermatocyte is known as primary spermatocyte, which will undergo meiotic division to form secondary spermatocyte, or we can say spermatocyte 2, SCC2. This spermatocyte will finally it will undergo the division to form spermatids. Now these spermatids are the structures which are without tail. Finally, these spermatids will be converted into spermatozoa. So all these process of formation of spermatozoa, which is nothing but these sperm with well-developed tail and everything, is this whole process of formation of spermatogonial mother cells is known from spermatogonial mother cells to the spermatozoa. This whole process is known as spermatogenesis. Then let us see the role of Sertoli cells. Now, Sertoli cells are the ones which are present in between the, we have seen here already, they are present in between the spermatogonial cells. They are the part of seminiferous tubule only, whereas the interstitial space was not the part of seminiferous tubule. They are the space which are present outside the seminiferous tubule. Now, you can see here spermatogonia. In between the spermatogonia, we can see the Sertoli cells. Now, what is the function of these Sertoli cells? Is nothing but they provide nutrition to the spermatozoa. So they provide nutrition to the spermatozoa and also produce a hormone known as inhibin. This inhibin hormones will be finally, the main function of this hormone inhibin, which is secreted from the Sertoli cells, is it inhibits the secretion of FSH, that is nothing but follicle stimulating hormone. So that is the main role of Sertoli cells here. There are two functions. One is it provides nutrition to the spermatozoa, and the next is it releases the hormone inhibin. So it is having endocrinal nature also. This inhibin inhibits, this hormone inhibits, the name is inhibin because it inhibits the secretion of FSH. The next thing is interstitial space. Now this interstitial space is the space which is present outside the seminiferous tubule. So the region which is outside the seminiferous tubule, if we take, this is one seminiferous tubule, this is another seminiferous tubule, this one is another seminiferous tubule. So the region which is present in between, or the space outside the seminiferous tubule, is known as interstitial space. Now this interstitial space consists of interstitial cells or cells of Leydig. So the cells which are present in this interstitial space are the Leydig cells or cells of Leydig. Now what is the role of these Leydig cells? Leydig cells produce androgens. Now one of the most important androgen is testosterone. And we know what is the role of testosterone. Testosterone controls the development of secondary sexual characters, and that we know what are the secondary sexual characters in males. So the main role of testosterone is to control the secondary sexual characters in males, as well as it also controls the process of spermatogenesis. So we know that the process of gametogenesis, whether oogenesis or spermatogenesis, they are under the hormonal control. So this start. The next thing is next structure is epididymis or epididymis. This epididymis, now here you can see clearly the vast difference. Vast difference where we have seen vast difference for nothing but the seminiferous tubules. This is the seminiferous tubule finally opening into the tube-like structure, which is known as retate testis. The end of the seminiferous tubules. This part is known as retate testis. These retate testis are finally opening into the vas difference. Okay. And these vas difference will be opening into the epididymis. So now what is the location of this epididymis and what exactly epididymis is a narrow, tightly coiled tube-like structures. These are narrow, tightly coiled tube-like structures, and they are located along the posterior surface of the each testis. So as we know, a pair of epididymis is present, and it is located on the posterior surface of each testis. Now, what is the role of this epididymis? The main function of this epididymis is it stores the sperm. It provides a storage space for the sperm and gives the sperm time to mature. So that these are the two functions of epididymis. Stores the sperm and it gives time for the sperm to mature. Now the epididymis is divided. This is the epididymis. It is divided into three parts. That is head of the epididymis, body of the epididymis, and the tail of the epididymis. So head of the epididymis is known as caput epididymis. Now the vas efferents opens into the head of the epididymis, and then the next part, that is it reaches to the body of the epididymis, which is the middle part of the epididymis known as corpus epididymis, and the last part of the epididymis is cauda epididymis, which is nothing but the tail of the epididymis. So head is the anterior most part, body of the epididymis is the middle part, and tail of the epididymis is the end part. Now this epididymis finally opens into the next structure, which is known as vas deferens. Now let us see here what is vas deferens or vas differentia, also known as ductus differentia. Now you can see the testis here, the testis which is present inside this is the scrotal sac. This is the scrotal sac. And inside this testis is descended. Now this testis will be containing seminiferous tubules which opens into the retate testis and then epididymis, and finally this epididymis is opening into the vas deferens. So this part which is coming out from the testis is the vas difference. You can see here, this is the vas difference. Now, this vas difference is a long, narrow muscular tube. Now, long, narrow and muscular tube means it is made up of muscles. So there will be muscles present here, which will be moving by the process of peristaltic movement. And it starts from the tail of the epididymis. So which was the end part of the epididymis, that is tail of the epididymis, leads to the next part, which is vas difference. And this vas difference will be passing through the inguinal canal. Yeah. Let's go to the slide. Here, see the inguinal canal. So the vas difference finally, when it epididymis, this vas difference to the abdominal cavity through this inguinal canal. So from here it will be coming out. Now, it passes through the inguinal canal into abdomen and it loops over the urinary bladder. Now, this part is the urinary bladder. This is the urinary bladder and it is looping over the urinary bladder. Now, on the way, this vas difference will be receiving a duct from the seminal vesicle. You can see here a pair of seminal vesicles. This is one and this is another one. A pair of seminal vesicles, which are nothing but the accessory glands of the male accessory glands. These seminal vesicles will be releasing its fluid. It is opening out through a duct into the vas difference. So, finally, the secretions of the seminal vesicles and vas difference are coming together, and these two, vas difference plus the duct of seminal vesicle, unite to form a short ejaculatory duct. And these both, the ducts of vas difference and seminal vesicle, which are forming the ejaculatory duct on both the sides. So this is one vas difference and seminal vesicle. This is another vas difference and seminal vesicle, which is finally opening into the prostate gland. Now this is they both will be converging into the prostate gland. This part is the prostate gland, and then it opens into the urethra. So vas difference is coming out of the inguinal canal by carrying the sperm, which vas difference got from the tail of the epididymis, and it opens into the urethra through the urethra along with the secretions from the seminal vesicle. So now this vas difference is having both the sperm plus the secretion of the seminal vesicle. Now in the next slide, we'll be discussing. Now this vas difference, it opens into the urethra, this part. This is the urethra, and we can see here the vas difference is opening into the urethra along with the ducts of seminal vesicle together forming the ejaculatory duct. Now, in male, the urethra shares terminal ducts of reproductive and urinary system. So both the reproductive duct and that is both the reproductive things, that is male germ cells, and the urinary system, that is which carries the urine, both this urethra is the common duct for both of them, reproductive system as well as urinary system, which is seen in the males. And this originates from the neck of the urinary bladder. Now you can see here, this is the end part of the urinary bladder. So from here, the urethra is originating. So this is the origin of urethra. And this originates from the neck of the urinary bladder and extends through the penis to external openings. So finally, it opens out to the outside through a structure known as urethral meatus. This is the opening to the outside and which is present at the tip of the penis. And it provides an exit for urine and semen during the ejaculation in the male. So this is the structure of urethra. So urethra will receive its urethra, either it will receive urine from the urinary bladder or it receives the sperm along with the seminal fluid from the vas deferens. So these are the two openings through which urethra will get its. The next structure is penis. Now penis serves as urinary duct, and the penis and the scrotum constitute the male external genitalia. The penis serves as a urinary duct and also an intromittent organ that transfers the sperm into the vagina of the female by the process of insemination. The human penis is made up of 3 columns of tissue. Okay, these 3 columns of tissues are nothing but 2 upper corpora cavernosa, which is dorsal in location, and the lower one is corpus spongiosum, which is ventral in location. So these are the 2 main types of tissues which will be forming the structure of penis: the corpora cavernosa, which is dorsal in location, and corpora spongiosum, which is ventral in location. Now, the skin and the subcutaneous layer enclose all these 3 columns. So these 2 upper corpora cavernosa and these 2 upper corpora cavernosa and corpus spongiosum, they are covered by skin, which is a subcutaneous skin and a subcutaneous layer. This corpora cavernosa consists of special tissues with spaces that are filled with blood. So that is the main role of this corpora cavernosa. The next thing is the enlarged and bulbous end of penis is known as glans penis, and it is covered by a loose fold of skin which is known as prepuce. So enlarged and bulbous end of the penis is known as glans penis, and this glans penis is covered by a loose fold of skin which is known as prepuce. And then this urethral meatus, which is nothing but the opening of the opening to the outside. This urethral meatus is the one which is lying at the tip of the glans penis. So this part you can see here, it is the glans penis, and opening of the glans penis is nothing but the urethral meatus, through which the secretions will be released out during the process of ejaculation in the male. So the urethra transverse the corpus spongiosum, and its opening called urethral meatus lies at the tip of the glans penis, which is the peripheral part of the male reproductive system. Now, the next thing is, what are the male accessory genital glands? So here there are 3 accessory genital glands in the male: a pair of seminal vesicle, 1 prostate gland, and a pair of pulbourethral gland. Now, if you want to see the location, the seminal vesicle is here, this, and then the prostate gland, and here we will find the bulbourethral gland just below the prostate gland. Now let's see about each and every accessory gland. The seminal vesicle, a pair of seminal vesicle is present in the male reproductive system. The location is posterior inferior to the urinary bladder. Now you see here, this is the urinary bladder. So this seminal vesicles are present on the posterior inferior side of the bladder. And it opens into the
Krishnavari,
vas
madam.
deferens. So finally,
Yes.
Conclude, madam.
Yes. చెప్పండి.
Madam, 11:35. Conclude.
Yes, yes.
okay.
She will.
So, okay. So next part we'll be discussing tomorrow. So I'm concluding the class. The male accessory glands will be discussing tomorrow.
Okay, thank you.
Thank you.